Hey yo, what's up, everyone? It's your boy JOSHABRAHAM, and we're back with the sequence analysis 1 hour. I'm gonna be only studying this for 1 hour. Let's see how much progress we can get within 1 hour. Let's here set a timer for 30 minutes. Okay. So this is the question, and from what I understood, it's Fitch algorithm. If you want, I can explain to you the topology of figure 1. Given the following imaginary multiple sequence alignment comprising 9 tags in the total. Human sequence, so till B subtilis is gonna start with R. Rice, rye, pig. Apply Fitch algorithm as discussed in the lecture. The imaginary tree topology depicted below using pen and paper. For this purpose, concentrate on the last column of the MSA. I don't think ChatGPT is gonna handle this very well. Explain Fitch algorithm clear up step by step. You can apply it to the tree. The example, compute the columns. Fitch algorithm. Compute the minimal number of substitutions, parsimony score for that column on a given unrooted tree. Inputs. A tree topology with tags at the leaves. You need exact topology to compute numbers. The character at the column for each leaf. For the last column, you gave the characters see below. Algorithm summary. Label leaves with. Okay, you know what? I think YouTube is gonna be a better choice. Six minutes, let me see. It's 15-second ads. Lord have mercy.
And we're going to look at how to use parsimony to evaluate phylogenetic trees. Okay? So, parsimony is the idea that the simplest explanation is usually the correct one. So, in the case of phylogenies, we assume that evolutionary events, evolutionary changes, happen relatively rarely. And so, a phylogenetic tree that requires fewer evolutionary changes is more likely to have occurred than a phylogenetic tree that requires more evolutionary changes. All right, so let's look how we can apply that to a specific phylogenetic tree. So these are the great apes, the old world apes. There are actually a few more species in this, but we're going to simplify it just kind of generally. We have chimpanzees, gorillas, orangutans, and humans, okay? So we want to know the evolutionary relationship between these organisms. And we're just going to focus on the chimpanzees, gorillas, and humans, and we're going to use orangutans as our outgroup because they're a more distantly related ape than the chimpanzees, the gorillas, and the humans, okay? So if we want to build trees that contain different relationships between chimpanzees, gorillas, and humans, with only 3 members in our in-group, there's actually only 3 trees you can build. You can either build a tree, let's take a look at these trees. You can either build a tree where humans are most closely related to chimpanzees.
Mm-hmm.
So that looks like this tree here. Humans and chimpanzees are more closely related. They're more distantly related to gorillas, and then you have your outgroup of the orangutans. You can build a tree where chimpanzees and gorillas are more closely related to each other, and then humans are more distantly related. Or you can build a tree where humans and gorillas are more closely related to each other, and chimpanzees are more distantly related. Okay, so those are your 3 possibilities, and those are the only 3 possibilities if you're using orangutans as an outgroup and you only have 3 species in your in-group. Okay, so these are our 3 trees. So how do we tell which tree is right? Or which tree has the best support from the information that we have? All right, so we're going to use to determine which tree has the best support, we're going to use some genetic data. So at the bottom here, we have globin pseudogene sequences. It doesn't really matter what that is. It's just some DNA that was sequenced for each of these species. And we have the sequence of those DNAs from the 4 organisms in question. Now, remember, orangutans are our outgroup, so we assume that they have the ancestral trait, and then we can look for shared derived traits within the in-group. So let's take a look at some examples. So in this scenario, here's a couple of sites that we might look at. Let's start by looking at this site here that is all T's. Does this tell you anything about the relationships between the different organisms in this group? They all have a T. It doesn't tell you which one's more closely related to which other one. So that doesn't really help us. This one here, you might think this one seems a little bit better because there's only humans have a T and the other 3 have C's, right? But actually, this doesn't really help you either because all of these guys have the ancestral trait and only humans have the derived trait. In order to build a tree, we need shared derived traits, not traits that only one organism has. And the reason why it doesn't really help is because if you go in here and you map where that T evolved, it evolved after humans separated from everything else. So it evolved here, or here, or here. But in every case, that's only one evolutionary event. It happens sometime after humans diverge from something else, but it doesn't help you to group them. So what we need are shared derived traits. So what does a shared derived trait look like? That looks like this. So here's a trait that is a shared derived trait. So in this scenario, humans and chimps share the G, which is derived because the ancestral trait is the T, okay? So gorillas have a T with the orangutans, and the humans and chimps have a G, okay? Now, let's map this trait onto each of the trees, okay? So assuming that the tree is true, where on the tree would this trait evolve, okay? So let's take a look at this. In the first tree here, you can have it evolve right here. And so humans and chimpanzees have the shared derived trait, and the gorillas and orangutans both have an ancestral trait. Okay, so it just requires one evolutionary change. Now let's try and put it onto this tree in the middle here. Now, the problem is that now gorillas are here. They're most closely related to chimpanzees, and humans are over here, right? So if you map the G's onto this tree, you see that you have to have it evolve twice. Because if it evolves down here, which is possible, it could evolve right here, but then it would have to switch back in the gorilla lineage, and that would still be two evolutionary changes. So it either has to evolve separately in humans and chimpanzees, or evolve in the whole group and then switch back in the gorillas. Either way, it's two evolutionary changes. Okay? And then for the last tree, similar scenario. Here I've illustrated the changing in the whole group and then changing back in the gorillas, but it's the same deal. You have two evolutionary changes. So if you're looking at these trees based on this one site within the genome, which tree is most parsimonious? Well, the first tree, tree number 1, only requires one evolutionary change, so it is the most parsimonious tree. Now, if you had a bunch of different data, which normally you do, you would collect a bunch of different traits and map them all onto the tree and see which tree requires the fewest evolutionary changes to occur. And then that is going to be your most parsimonious tree. So that's how we use parsimony to determine which is the best tree.
Mm-hmm. Oh, that's it. Okay, I kinda understood, but how do I... Imaginary tree topology depicted below using pen and paper. For this purpose, concentrate on the last column of the MSA. What do you mean by MSA? Explain each of the intermediate steps necessary to compute the corresponding tree length. Does it make sense to apply Fitch's algorithm to other columns of the current MSA? Discuss your answer in detail using the same multiple sequence MSA multiple sequence alignment as defined above. Calculate the corresponding entropy of the entropy for each column. Entropy? What? What entropy? What the?
Okay, so under maximum parsimony, the best tree is the shortest tree, the tree requiring the fewest mutations. The question is, how do we, for any given data set, find the maximum parsimony tree? How do we find the best tree? Topologies all the possible things that we can think of for this particular size data set. Then, for each tree, we try to determine, okay, on this particular tree, how many mutations are required by our data, just like we did in the previous lecture. On this particular tree, how many mutations would be required for this particular data set. When we're finished, when we've looked at all the possible trees, we just select the shortest one from the list. If it should turn out that there are more than one tree that have the same length, then they are, by definition, equally good, equally parsimonious. We have no way of choosing between them under this particular criterion. Okay, so this sounds like a reasonable approach to finding a parsimony tree, but it of course leaves us with a new set of sub-problems. In particular, first of all, we need some algorithm for how to construct a list of possible trees. How do we do that? How do we, for a data set of 20 organisms, write up all the possible trees, for instance? Secondly, we need an algorithm for determining the length, the number of required mutations for any given tree. I mean, we could do it by hand before, just by sort of looking at the sequence, saying, oh, we need a T here and a G here. But if we have a large data set, this will, of course, be impossible. So we need some sort of computer algorithm that, for a given tree and a given data set, can tell us how many mutations are required on this tree. So let's just look at how we solve these two sub-problems. First, constructing the list of all possible unrooted, as it were, trees. How do we do that? It turns out that a very simple algorithm is to gradually build the tree one leaf at a time. If we start out, as indicated on this slide, by building a tree with only 3 species, then there is just one way of doing that. So we just pick the first 3 species in our data set and construct a phylogenetic tree from that. It will have A, B, and C sticking out. There's no grouping, there's nothing ambivalent about that. Starting with that tree, we can now add the 4th species. As it turns out, we can add the fourth species in 3 different places. And we can add the fourth species in 3 different places because there are 3 branches on the initial tree. So we could put species D, we could put that on the B branch, on the C branch, or on the A branch. Okay? So for each branch in the original tree, we can add the next species, giving us, in this case, 3 derived trees with 4 species. From each of these, we can now add the 5th species, and it's the same principle again. On this particular tree with 4 species, we can now add species E in 5 different places because there are 5 branches on this tree. So we could add it on the A branch, on the B branch, on the C branch, or on the D branch, or in the central branch, giving us a total of 5 possible derived trees. We could, of course, do the same for the 2 other starting trees. So each time we have a starting tree, we can add the next species to each branch. Then on that resulting tree, we can add the next species on each branch, etc., until at some point we hit the total number of species, and at that point we will have the list of all possible trees. So problem number 1 solved, subproblem number 1 solved. We actually have a simple algorithm for how to build the list of all possible tree topologies. How about finding the length of a given tree? Well, that is an algorithm that also exists. There are a number of ways of doing it. A very important algorithm for doing this is called the Fitch algorithm, and that's what I'm going to explain to you. It's a very simple algorithm. It's the type of thing you can do in the back of an envelope if you're really bored and trying to make a maximum parsimony tree. Okay, so here's a tree. We have just a single nucleotide to keep things simple, but if you have more nucleotides, you just add it up for every nucleotide position. So this is essentially a 1 nucleotide alignment. We have A, C, C, A, G, and they're located on the tree in this particular manner. This is not the most parsimonious tree, incidentally, for this particular data set. It's just to show you how you would compute the length of this particular tree, because as I said, we have to compare all possible trees for any given data set. But how many mutational steps are required for this? Well, here's the algorithm. I'm just going to put it up. I'm going to read it out to you, and then we're going to walk through an example of how that works. The idea in the Fitch algorithm is that you start out by rooting the tree at some arbitrary internal node or some arbitrary internal branch. Secondly, this now gives you a rooted tree where there's a direction where you have parents and descendants. You then start by visiting some internal node X, for which we have no state set, I'll return to what that is, but where we know the state set of its immediate descendants. So a state set could be a number of possible nucleotides. So for a leaf, there is just a single nucleotide, namely the nucleotide that's actually present there. So an internal node where we don't know the state set, but where we know the state set of its descendants, would be an internal node having 2 leaves attached to it. Okay. If these descendants, if the state sets in these descendants have something in common, then we assign that to X. If they do not have anything in common, if these 2 descendants have no common states, then we assign the union, the totality of all the states that they have, to our internal node X, and we increase the tree length by 1. I'll explain this on the tree again also. We then repeat this, going down the tree, walking down internal nodes, adding possible state sets to all internal nodes until we reach the node. At this point, the length of the current tree is whatever it was at that point. Remember, we increased it by 1 every time we had to assign the union here. So let's just walk through an example so this will hopefully become clear. So we start out with our tree. We now pick an arbitrary internal branch to root it on. So let's say we place the root on this internal branch. So I pull down on this internal branch and say this is now the root. I then fold up the rest of the nucleotides so we get this rooted tree. So this is the same tree as we started out with. I just pulled down the internal branch there and folded up the nucleotides. Okay. Step number 1 in the algorithm, this was step number 1. Step number 2 in the algorithm is now to visit an internal node where we don't know the state set, but where the state set of its immediate descendants have been defined. So one example is the node over here at the left, where the descendants are C and A. So the descendants have state set C and state set A. They have nothing in common. Obviously, they're 2 different nucleotides, and we therefore have to assign the union of these nucleotides to the internal node. This has a C, this has an A, they have nothing in common. We assign the union to the internal node, and we increase the tree length by 1. So the length so far is 1. Why is the length 1? Well, what we're doing here is actually we're starting to think about what the ancestral nucleotides could be in a manner of speaking. Okay, so this ancestor, we now say, was probably either a C or an A. If it was a C, then there were 0 mutations on that branch, but there must have been a mutation on the branch leading to A. If the ancestor was an A, then there were 0 mutations on the A branch, but there has been a mutation on the C branch. So no matter what state this ancestor had, there must have been 1 mutation corresponding to a tree length of 1 so far for this subtree. Okay, so let's move to the other side of the tree. Again, there's an internal node over here with the A and the G. Again, they have nothing in common, so we have to assign...
Just a minute.
union A and G to that internal node, we have to increase the tree length by 1. No matter what the ancestral nucleotide was here, there must have been 1 mutation. We now get to this internal node. One of the descendants of this internal node is a leaf with a C. The other descendant is an internal node with state set A and G.
I need to see this once more. I still haven't seen the algorithm. Like,
Again, they have nothing in common, and we have to assign the union A, C, and G to that and increase the tree length for the same reason that we did before. We're now down to the root. One descendant is an internal node with A and C. The other descendant is an internal node with A, C, and G. Now they do have something in common. They have A and C in common. So we assign that to the root. We didn't have to increase the tree length. We're done with the algorithm, and we can now note that the total length of this tree must be 3. One way of seeing that this is true is to actually try to put in, to make one ancestral reconstruction and see that the number of mutations fit. So one possible reconstruction, there are actually other possibilities, is this. We assume that the ancestor was an A and that all the other ancestral nucleotides were also A. If that's true, then we can see there's been one mutation on the branch to the C, another mutation on this branch to the C, and one mutation on the branch to the G. So all in all, this particular tree with this particular data set, 3 mutations are required. That's what the algorithm was supposed to tell us. And you can see that we could do that for any given data set for any given tree. So both of our subproblems in solving the maximum parsimony problem have now been solved.
So Fitch's algorithm, how to apply step by step. For the last column, compute the minimal number of substitutions for the columns on a given unrooted tree. Input a tree topology with tax algorithm summary. Label leaves with the absurd character. Post order pass. Compute sets the internal node. S is not empty. S node optional pre-order. Pass to assign specific states to the nodes. If you want to reconstruct one possible character assignment, intersections non-empty means both children can share the same state with no additional substitution. How to apply on. Write the leaf characters at each leaf. I don't get that. Choose an internal node. Write 2 child sets. Computes intersection union whether you do 0 or +1. Mark the node set. Move up until the root. Sum all add +1's parsimony score for that column. When the instructor asks you concentrate on the last column, do exactly this on the paper. Okay, I still didn't quite understood how to proceed with this. Like, where do I start? Sorry, I ain't got no money. I'm trying to be funny. I'm today. It's time. You do not start with entropy. You do not start with the whole MSA. You start with one column and one tree. Just a minute. 30 minutes? God damn! Fitch algorithm always starts with the tree topology characters at the leaves at one column. Your assignment says apply Fitch algorithm to the last column, so we must write down the character at the last column for each taxon. C, D, C, C, okay. Now, so the leaf states are, for one, it's C, okay. Draw the tree from the exam. The figure is basically this grouping, you described it earlier. 1, 4, oh, 3. The 6 attaches to the very bottom. I thought 8 was the very bottom now. This matches the ordering assignment. Okay. We start from a common point. We diverge 2 ways. And let's take the left side. Left side, we diverge again to 2 ways, which goes to a point where it's divided. It's if it went straight, it's 9. If it had a mutation, it's gonna be 2. Now the left part, again 2 ways. Straight, if we went the straight way, it's gonna be 3. If we went to the left, it's gonna again divide into 2 parts, 1, 4. Can you do a Python? Explain each of the intermediate step necessary to compute the corresponding tree length. Does it make sense to apply Fitch algorithm to other columns of the current MSA? Discuss your answer in detail. Again, I have the figure on the question itself. I didn't understand what's the starting point, and others like wait, so 1 and 4, 1 and 4, 1 and 4, that's the C, right? So they both are C. And that what's 3? 3 is also C. Uh-huh. One, 4, and 3. All these are C. Then why is 3 diverged from the common of 1 and 4? Read good. That this tells me exactly what you're stuck on. You're confused on how to read the tree in the figure. This means you're misreading the topology. Let me explain how the tree in the assignment is actually structured. The actual tree structure based on the figure the assignments list on taxa 2. Why is 6 up here? 6 is here. This is not a simple left to right wrist, it's an unrooted tree drawn sideways. What is 4? What is 9? It's N. What is 2? That's E. Yes, there's a timer for 15 minutes. I want to describe this tree to ChatGPT. Okay. I'll describe how the tree looks. You are only seeing the last part. So first there is a common point or starting point, then it diverges to 2 points with the right side again diverges to the number 5, then after another diverge to 7, again another 6, and at last it ends at 8. Now the initial part that diverges to left first diverge at to a common point, which again diverge to 2 paths with 2 on the left and 9 on the right. Is straight to 3, then the next What? Eight, 6, 7, 5. Yeah. Oh, yeah, that's basically a 3. 5 to 7. I don't get it fully, but I'm starting to understand something. Oh man. Again, I lost my focus, I guess. Attention span. Oh. Yep, I'm gonna end the stream, take a small break, continue with this biochemical kinetics. Then after that, I'm gonna continue with intro to general chemistry. Yep, I don't want to waste time. I'm gonna take a small break and come back. Peace out, YouTube.